Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Welcome back to the deep dive. I want to start today by asking you to visualize something that feels, well, almost impossible to solve.
Okay.
Imagine a city where the sanitation department has gone on strike, but the factories are still running at full capacity.
Right? I can picture that.
So the trash is piling up on the sidewalks, then it blocks the streets, and eventually it starts breaking through the windows of the city hall, physically stopping the government from working. Now, take that image and shrink it down until it fits inside a single microscopic cell in the human brain.
And that's where we're headed today.
We are standing at the edge of a very steep cliff today, looking down into one of the most complex and heartbreaking biological puzzles humanity has ever faced. Huntington's disease.
It's a profound analogy, and unfortunately, it is entirely accurate. Huntington's is often described in textbooks as a genetic disorder, which it is, but functionally, it's a waste management crisis.
Waste management crisis.
Exactly. It's a fatal neurodegenerative condition that it erodes a person's physical control and their cognitive self. And despite decades of brilliant people working on it, we currently have no cure.
No cure.
We can manage some symptoms, but we can't stop the progression.
That is the heavy reality we are dealing with. And I want to be very clear about this right from the start.
Yeah.
We are not here today just to admire the problem.
No.
We're here because of a specific massive piece of research that was released just this month, February 2026.
Oh.
It comes from a team of heavy hitters in the scientific world, and it might just have handed us a key to the city sanitation department.
This is a significant pivot in how we think about the disease. Usually the conversation focuses on the production side of the toxic protein. We're always asking how to stop the factory.
Right.
This new research asks a different question. It asks, what if the problem is that we just lost the address label for the trash can?
That is the core premise we are exploring, the trash collector's dilemma. To guide us through this, we're pulling from a stack of primary sources. We have a news release from Medical Express dated February 8, 2026, and then the heavy-hitting scientific paper itself.
Published in the Proceedings of the National Academy of Sciences, PNAS, in January 2026.
Right, and the research was led by Professor Hufok Nguyen at Ruhr University Bochum. But there's another name on this paper that made me sit up straight.
Professor Aaron Ciechanover.
Exactly. For those who don't, track Nobel Prize winners like baseball stats, why is his involvement so important here?
Well, Aaron Ciechanover won the Nobel Prize in Chemistry back in 2004. He didn't just win it for any discovery. He won it specifically for discovering the ubiquitin-mediated protein degradation system.
So he literally discovered the cellular trash can.
He discovered the entire system, the tags, the process, the machinery. So when you have the man who wrote the rules of protein disposal, co-authoring a paper about a disease defined by protein accumulation, you know the methodology is going to be incredibly rigorous.
It adds a whole other layer of gravity to the findings. So our mission today is to unpack this study. We're going to zoom in on two tiny microscopic spots on a protein, literally just two amino acids, and see how they might be the difference between a healthy brain cell and cell death.
It really is a detective story that takes place on the molecular level.
And if we follow the clues correctly,
It leads us to a very hopeful discussion about the future of therapy.
Okay, so let's start with the antagonist of our story. Before we can understand the solution, we have to understand the problem. We need to talk about mutant Huntingtin.
Right. To understand the mutant, we first have to understand the original. Every human being has the HTT gene. It produces a protein called Huntingtin. We all have it, and it's actually essential for normal brain development.
So the protein itself isn't the bad guy.
Not at all. The issue arises from a kind of stutter in the genetic code.
A typo in the blueprint.
Exactly. The DNA code uses 4 letters, and in this gene there's a sequence of 3 letters, cytosine, adenine, guanine, or CAG, that repeats.
CAG over and over.
Yes. And in a healthy gene, this CAG sequence might repeat, say, 10 to 75 times. It creates a short tail of an amino acid called glutamine on the protein, no problem.
But in Huntington's disease, that stutter gets out of control.
The repeat expands. It goes over 36 and sometimes way up into the hundreds. Since the DNA is the blueprint, the cell's factory builds the protein exactly as written.
With this giant tail.
The resulting Huntingtin protein has a massive elongated tail of polyglutamine, and that tail is the problem. It changes the physics of the protein.
It causes it to misfold.
It causes misfolding. And in the world of proteins, structure is function. If you fold correctly, you're a machine that does a job. If you fold incorrectly, you are garbage.
Or worse.
Or worse, you're sticky garbage. This mutant Huntingtin protein tends to clump together, forming what we call aggregates. These clumps are toxic. They trap other important proteins. They gum up the cellular machinery, and eventually they kill the cell.
Yeah. Okay, so we have the factory churning out these broken, sticky widgets. Now, in a healthy cell, this shouldn't be a catastrophe. The cell has a quality control department.
And this is where a protagonist enters the story, Ubiquitin.
I love that name. It sounds like it's everywhere.
That's literally the etymology. It's ubiquitous. Ubiquitin is a small regulatory protein found in almost all tissues. Think of it as a physical tag.
Like a red tag on a broken piece of equipment.
A red tag or a sticky note. When the cell identifies a protein that is broken, misfolded, or just no longer needed, enzymes attach a chain of these ubiquitin molecules to it.
So it's like putting a sticker on a broken chair that says, "To the dump."
Precisely. And that tag is a signal for the proteasome. The proteasome is this massive molecular machine. Think of it as a barrel-shaped industrial shredder.
Wow.
It just floats around the cell looking for these ubiquitin tags. If it sees the tag, it grabs the protein, pulls it inside the barrel, and shreds it back down into amino acids to be recycled.
This is the ubiquitin proteasome system. So in a perfect world, the mutant Huntingtin gets made, the cell sees it's misfolded, slaps a ubiquitin tag on it, and the proteasome destroys it. Crisis averted.
That is how it should work. But in Huntington's disease, clearly it's not working. The trash is piling up. The aggregates are forming. The question this study sought to answer was, why?
Is the shredder broken?
Is the shredder broken? Is the tag not sticky enough? Or, and this is the crucial hypothesis, are we putting the tag in the wrong place?
This brings us to the specific mystery of K6 and K9. These sound like coordinates on a map.
They are coordinates on the protein map. Proteins are long chains of amino acids. Scientists use the letter K to represent the amino acid lysine.
K for lysine. Okay.
So K6 refers to the lysine residue at position number 6 on the Huntingtin protein chain, and K9 is the lysine at position number 9.
And since the protein can be thousands of units long, position 6 and 9 are right at the very beginning, like the front door of the protein.
We call it the N-terminus. And the researchers believe that these two specific spots, K6 and K9, are not just random parking spots for the tag. They hypothesize that these are the essential addresses.
The essential ones.
They suspected that for the Huntingtin protein to be properly destroyed, the ubiquitin tag must be attached specifically at position 6 and position 9.
So if you tag it somewhere else on the protein, the janitor might just ignore it.
Or the process becomes incredibly inefficient. The hypothesis was that K6 and K9 are the master switches for the disposal of this specific protein.
How do you even test something that specific? You can't just, interview a cell.
No, you have to force the biology to reveal its secrets. This leads us to the experimental design, which I found fascinating. They didn't just use a petri dish. They engineered a very specific type of mouse.
Okay.
They used what is called a knock-in mouse model. This is a crucial distinction.
Oh.
A lot of studies use transgenic mice, where you just inject the human gene into the mouse embryo. So the mouse has its own genes plus the human disease gene.
It's like it's wearing a backpack with a bad gene in it.
Right. But a knock-in is much more precise. They actually went into the mouse's DNA and replaced the mouse Huntingtin gene with the human mutant version.
So the mouse is running the human software natively.
Exactly. And they created 2 teams of these mice to battle it out.
The control group and the experimental group.
Let's call them that. The control group was named the Q134KK mice.
Okay, let's decode that. Q134.
Q stands for glutamine. 134 means there are 134 repeats of that CAG stutter. That is a massive mutation.
In a human, that would be very bad.
Very severe, probably juvenile onset of the disease. So we know these mice are genetically destined to get very sick.
And the KK at the end.
The KK means that at positions 6 and 9, the amino acids are lysine K. They're normal. They can accept a ubiquitin tag. So this group represents standard severe Huntington's disease.
Okay, so that's our baseline. Now for the experimental group, the Q134RR mice.
They have the same toxic mutation 134 repeats. But, and this is the brilliant part, the researchers used genetic engineering to change position 6 and position 9 from lysine K to arginine.
Why change it to arginine? What's special about that?
Because arginine is chemically very similar to lysine. It has a similar shape, a similar charge, so it doesn't break the protein's overall structure. However,
There's the catch.
the key is arginine cannot bond with ubiquitin. You physically cannot attach the tag to it.
So they created a protein that is effectively invisible to the specific trash collectors looking for those spots.
They removed the address labels. That's it.
So here's the setup.
Mhm.
Both groups have the fatal disease gene.
Hmm.
The only difference is that one group cannot be tagged at these two tiny spots if the hypothesis is wrong.
The mice should look more or less the same. The disease should progress similarly.
But if the hypothesis is right,
If the hypothesis is right and those tags are critical for disposal, then the mice without them should get much, much worse.
and that is exactly what happened. Let's walk through the findings, because worse is an understatement. The paper describes a system failure.
It was comprehensive. I mean, let's just look at the physical toll first. One of the primary indicators of health in these mouse models is body weight.
Right.
If the metabolism is failing, the animal loses weight. The no-tag mice, the Q134RR group, showed a drastic decline in body weight, starting at around 6 months of age.
While the control mice, the ones who still had the disease but could be tagged, they were more stable.
They declined, but much, much slower. The no-tag mice were just wasting away. But the motor symptoms were even more telling.
You mean beyond the weight loss?
Yes. They performed a test called the clasping test.
Explain that to us. It's a very visual test from what I've read.
It is. It relies on a mouse's natural reflex. If you lift a healthy mouse by its tail, it will splay its legs out wide, reaching for the ground. It's a ready-for-action posture.
Right, trying to orient itself.
But if a mouse has neurological damage in the striatum, which is the part of the brain Huntington's really affects, it does the opposite. It pulls its limbs in. It sort of hugs itself.
It clasps.
It clasps. It's a classic sign of motor dysfunction and loss of inhibition. The mice without the K6 and K9 tags showed this clasping behavior significantly earlier and more severely than the control group.
So they were losing motor control faster.
Much faster. They also had an unsteady gait. They were taking shorter steps. They had tremors. They were physically falling apart.
There was also a finding regarding their sleep patterns that I think is incredibly relevant to human patients.
Oh, yes, the circadian rhythm disruption. They monitored these mice 247. Mice are nocturnal. They should be active at night and sleeping during the day. The no-tag mice became hyperactive during the day. They couldn't settle down. They were pacing constantly, moving with these fine tremors, even during their normal rest period.
That sounds just like the agitation and insomnia we see in human neurodegenerative patients.
It is remarkably similar. It suggests that the damage wasn't just in the motor centers, but deep in the regulatory centers of the brain, like the hypothalamus.
But the finding that really hit me the hardest was the cognitive one. How do you measure cognitive decline in a mouse?
You ask them to build a home.
The nesting test.
The nesting test. It's a brilliant measure of what we call executive function. You give a mouse some nesting material, usually it's a pressed cotton square, and you leave them overnight.
And a healthy mouse will do what with it?
A healthy mouse, or even a mouse with mild disease, has an instinct to shred that cotton and build a complex, structured, fluffy nest. It requires planning, dexterity, and motivation.
It's a project for them.
It is a project.
Yeah.
The no-tag mice, they failed at this project completely. Starting at just 5 months old, they lost the ability to build nests.
What did they do instead?
The cotton was just left scattered. Or maybe barely touched, they didn't know what to do with it, or they couldn't coordinate the action to do it.
And the control group?
The control mice could still build decent nests until they were much, much older.
So removing those two little tags destroyed their ability to plan and execute a task months before the disease naturally would have.
It confirms that the cognitive decline is tightly linked to the accumulation of this protein, specifically when the disposal system fails at these critical sites.
So physically and mentally, the no-tag mice were devastated. But as we know, symptoms are just the smoke. The fire is inside the cell.
Right.
The researchers then cracked open the cells to see what was happening. And part 4 of our deep dive is where things get really interesting. They call it the nuclear option.
This is the mechanistic breakthrough of the paper. We know mutant Huntingtin forms clumps, or aggregates, but where it clumps matters immensely.
Yeah. Location, location, location.
Absolutely. In the control mice, the Q134KK group, the ones that could still be tagged, the protein clumps were found mostly in the cytoplasm.
Which is the main body of the cell outside the nucleus.
Exactly. There were some in the nucleus, sure, but they were generally smaller.
But in the no-tag mice?
In the Q134RR mice, the protein aggregates were found almost exclusively inside the nucleus. And we're not talking about small specks. They formed massive, sharply defined structures called inclusion bodies.
The nucleus is the command center. That's where the DNA is kept. You do not want the trash piling up in the library where all the blueprints are stored.
You absolutely do not. This leads to the theory that the K6 and K9 ubiquitin tags act as a kind of keep-out sign for the nucleus.
Or maybe an export visa.
That's another way to think of it. If the protein is tagged, the cell knows to keep it in the cytoplasm and get it to the shredder. But if you cannot tag it, the protein slips past the security guards, enters the nucleus, and starts to grow.
And once it's in the nucleus, it's not just taking up space, it's actively sabotaging operations.
It causes what's called transcriptional dysregulation. Think of the nucleus as that library where the cell goes to read instructions on how to operate. The mutant Huntingtin is sticky. When it gets into the library, it starts sticking to the transcription factors, the librarians, preventing them from doing their job. It gums up the pages of the books.
So the cell forgets how to be a cell.
Essentially. The study showed that thousands of genes were being turned on or off incorrectly in the no-tag mice. The genetic confusion was far, far worse than in the control group.
There's a really specific detail about the clumps themselves that I want to verify with you. The paper talked about something called K48-linked ubiquitin.
Ah, yes. This is the smoking gun for the whole disposal theory.
Explain that code, K48.
Okay, so ubiquitin isn't just a single flag. It forms chains. And the way the chain is linked tells the cell what to do. A chain linked at a specific spot on the ubiquitin molecule, lysine 48 or K48, that is the universal signal for immediate destruction.
It's the death sentence.
It's the death sentence. If the proteasome sees a K48 chain, the shredder is on its way, no questions asked.
And what did they find in the no-tag mice?
When the researchers stained those clumps, specifically those large clumps inside the nucleus, they found they were almost completely devoid of K48 ubiquitin.
So without the K6 and K9 anchor points, the cell physically couldn't stamp the death sentence on the protein.
The disposal system was blind to them. They were completely ignored, so they just sat there in the most sensitive part of the cell, growing unchecked.
That explains everything. It explains why the symptoms were so much worse. The trash wasn't just piling up, it was piling up in the Oval Office, and the security team couldn't even see it.
And this brings us to another fascinating detail, something about RNA splicing. The study found that in the no-tag mice, the Huntingtin RNA itself was being processed differently.
How so?
The machinery that reads the instructions was so messed up that it created shorter, even more toxic fragments of the protein. It's a vicious cycle. The protein accumulates, it messes up the machinery that reads the instructions, which then causes even more toxic protein to be made.
It feeds on itself, a neurodegenerative cascade. I want to play devil's advocate for a moment here because we're putting a lot of weight on ubiquitin. But the scientist in you has to ask, is it definitely ubiquitin that attaches to K6 and K9? Or could it be something else?
That's a fair and a necessary scientific question, and the authors address it. The issue is that lysine residues, those K-spots, they're popular parking spots in the molecular world.
Prime real estate.
Very prime real estate. Ubiquitin isn't the only thing that attaches there.
So who else is fighting for this spot?
There are two main competitors. Summoylation, which is a small ubiquitin-like modifier, and acetylation. Both of these are also chemical modifications that attach to lysines.
So when the researchers changed K to R, they didn't just block ubiquitin.
They blocked all of them. They blocked SUMO and acetylation too. So theoretically, the damage could be because the protein wasn't getting, say, SUMOylated?
Theoretically, yes. However, the evidence leans very heavily toward ubiquitin. Acetylation usually does things like push proteins into the nucleus or help stabilize them. Since we saw the protein accumulate in the nucleus when the tag was blocked, it suggests that the missing tag was likely the one that keeps it out or destroys it. That points directly to ubiquitin.
Plus, the lack of those K48 signals, the death sentence signals, strongly implies the disposal system is the one that failed.
Correct. It's the most likely suspect, even if there might be other accomplices. And frankly, regardless of which specific tag it is, the actionable fact remains. Modifying K6 and K9 is the lever that controls the severity of the disease. Okay, so let's move to part 5. The implications. We have a mouse model. We have a mechanism. We know K6 and K9 are the control points. So what? What does this mean for a human patient?
The logic is wonderfully straightforward. If removing the tags makes the disease worse,
then enhancing the tagging at these sites could make the disease better.
Exactly. We want to supercharge the janitor. We want to promote ubiquitination at K6 and K9. This connects to a huge, exciting trend in pharmacology right now called targeted protein degradation.
This is different from gene silencing, right?
Yes. Gene silencing tries to stop the protein from being made in the first place. Targeted protein degradation accepts that the protein is there, but tries to clear it out faster. And the paper actually references a specific molecule, desonide.
Desonide, is that a new experimental drug?
Actually, no. It's a corticosteroid that has been around for a long, long time, usually for skin conditions. But some previous research showed it can promote the tagging of Huntingtin right at the N-terminus, where K6 is.
So now that we know for sure how critical K6 is.
Exactly. Now, drugs like desonide become very interesting starting points for new development.
But I imagine we can do better than an old skin cream.
Absolutely. This opens the door for a class of drugs called PROTACs.
PROTACs, I love this acronym. Break it down for me.
Proteolysis targeting chimeras. It's a mouthful, but the concept is brilliant. Imagine a drug that looks like a tiny dumbbell. One end of the dumbbell is designed to grab onto the toxic Huntingtin protein.
Okay.
The other end is designed to grab the cell's E3 ligase. That's the machine that actually applies the ubiquitin tag.
So the drug acts like a matchmaker. It physically drags the trash and the tagger together and forces them to hold hands.
That's a perfect analogy. It forces the ubiquitination to happen. And this study gives us the map. It tells the drug developers, hey, make sure your PROTAC exposes K6 and K9 so they can be tagged efficiently.
If we can force that tag onto the protein before it gets into the nucleus,
We might be able to prevent the whole cascade from starting. We could potentially convert a fatal, rapid decline into a much more manageable, slower condition. We keep the nucleus clean, we keep the library open for business.
it shifts the mindset from how do we fix the DNA, which is incredibly hard, to how do we help the cell clean up the mess.
And the beauty of it is that the cell wants to clean up the mess. The machinery is all there, the proteasome is waiting. We just need to help it print the labels correctly.
It feels like a more harmonious approach to medicine. You're helping the body do what it already evolved to do.
I agree. It leverages the body's own sophistication instead of trying to fight it.
So let's wrap this up. We started with what feels like an unsolvable puzzle, a genetic error that seemed to doom the brain. We looked at a study that took that error and made it untaggable in mice.
And by doing so, the researchers revealed the Achilles heel of the mutant protein. We learned that lysine 6 and lysine 9 are the essential anchor points. Without them, the protein invades the nucleus, disrupts the genetic code, and leads to a rapid, catastrophic collapse.
The Q134RR mice tell us a tragic story, but they leave us with a blueprint for hope. If we can develop drugs that ensure those tags get placed, drugs that act like super-efficient staplers, we have a real shot at a new kind of therapy.
It is a long road from a mouse model to a human cure, but having the precise coordinates of the failure point is a massive step forward.
For everyone listening, I want you to think about that next time you hear about genetic research. It's not always about splicing genes. Sometimes it's about understanding the microscopic traffic laws of the cell, who gets to go where, and who gets taken out to the trash.
And recognizing that even inside a single cell, there's a complex society of workers. And our job is to give them the tools they need to keep the factory running.
Absolutely. Thank you for joining us on this deep dive into the molecular machinery of Huntington's disease. It's complex, it's heavy, but it is also, well, incredibly fascinating.
Always a pleasure to explore the details.
Keep those neurons firing, stay
Mice.
curious, and we will see you on the next deep dive. Take care.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.